Okay, so welcome to this next video in which we are discussing reduced SERCA2 function in pathological cardiac hypertrophy. Okay, so what we're going to discuss now is the fact that phospholamban activity goes up in concentrically hypertrophied cardiomyocytes. So let me remind you of what phospholamban is. So we'll go on to the next page. So, phospholamban then. Phospholamban is a protein which is in the membrane of the sarcoplasmic reticulum, like the SERCA pump itself and like the type 2 ryanodine receptors. It's also often denoted as PLB for short, or also, confusingly, PLN. Both of those abbreviations are used for phospholamban. I will use the PLN abbreviation in this video. Right, so phospholamban is a protein which spans the membrane once. So if this is the phospholipid bilayer of the sarcoplasmic reticulum, then it spans the membrane once. However, it does not go around on its own. Instead, it goes around in pentamers. So 5 phospholamban proteins all associate together like so, 1, 2, 3, 4, 5. And they make a pentamer. So I'll draw these all in blue. So this is our phospholamban pentamer, which is in the membrane of the SR. So this is a pentamer. Right, now the job of phospholamban is to inhibit the SERCA pump. So if we draw a SERCA pump over here, then the role of phospholamban is to go and bind to the SERCA pump over here and stop it from functioning. So this is SERCA2. Okay? So it modulates the role of SERCA by inhibiting it, okay? And SERCA's role, remember, is to return the calcium back into the SR. So calcium, 2 calciums come in, 3 protons go out, and every time you do that, you are hydrolyzing ATP to ADP, an inorganic phosphate. So here's ATP going to ADP, and inorganic phosphate. Right, okay, so we know that SERCA2's function is too low in concentrically hypertrophied cardiomyocytes. So we might guess that maybe phospholamban activity is too high. And indeed what you find is that that is the case. In concentrically hypertrophied cardiomyocytes, what you find is that you have far too much phospholamban. So the phospholamban PLN expression is too high. So we've already seen that one of the reasons underlying SERCA2's downregulation in function is that you have just too little of it in the SR membrane. But now the stuff that you do have in the membrane is being inhibited by the too high amount of phospholamban that you have in there. Okay, right. In addition, I need to explain one final bit of this physiological pathway, which is that protein kinase A, okay, protein kinase A is capable of phosphorylating, okay, so let's stick a phosphate group on here. It's capable of phosphorylating phospholamban, and it will phosphorylate all of them, not just one, but I'll show it like just one. So it will phosphorylate phospholamban and stop it from binding to the SERCA2A. So by adding this phosphorylation onto phospholamban, protein kinase A can stop the phospholamban binding to SERCA2, and then if you stop phospholamban binding to SERCA2, then SERCA2's function is going to go up. So this is how protein kinase A increases the function of SERCA2 basically, by phosphorylating and inhibiting the phospholamban pentamers. Okay, right. So, what you also find is that not only is the actual amount of phospholamban that you have in the SR membrane too high, but you find that also the phosphorylation state of it is too low. So, you've got too much of it, and not enough of it is inactivated. Okay? So, you've got too much of it, it's overactive, and it's acting on SERCA2 pumps, which are already limited because their expression level is low. So all of these things are contributing to reducing the SERCA2 activity within the cardiomyocytes. So just to review, SERCA2's function is too low within concentrically hypertrophied cardiomyocytes. One of the reasons for that is that you just have too few SERCA2 pumps in the membrane of the SR. Another reason is that the SERCA2 pumps that are there are being inhibited by the phospholamban. In the concentrically hypertrophied cells, you have too higher expression of phospholamban, and this phospholamban is not very much phosphorylated. So it's basically, you've got too much phospholamban, and the phospholamban you do have is too active, so they're going to inhibit the SERCA2 pumps that are there, and therefore you're further going to reduce the function of SERCA2. So you're reducing the amount of calcium that you pump into the SR lumen, which means that the amount of calcium that's going to be in the SR lumen when the heart next beats is going to be lower. So the amount of calcium you can release in the calcium sparks is going to be reduced. So the size of the calcium tide you get, which remember is the sum of all the calcium sparks, is going to be reduced. And then if you've got a smaller calcium tide, then the number of sarcomeres you can activate to contract is going to be reduced. So the amount of force you produce is going to be reduced because you've got fewer sarcomeres actually contracting. Okay, right. One last little experiment to provide, one little fact rather, to provide proof of principle, which is that if you mutate the phospholamban gene, specifically if you mutate Arginine 14, so R is the single letter amino acid code for arginine, and we're talking about the arginine at the 14th position of the polypeptide making up phospholamban. So just for a little bit of added information, I'll show you the structure of arginine. So let's draw the core amino acid structure to start with. So here's the alpha carbon, the amino group, the carboxylic acid group down here, and then the R group of arginine is that you have these free methylene groups, which, because I'm lazy, I'll just put brackets around 1 and put 3 down there. Then you have a nitrogen coming off that, and then a carbon coming off the nitrogen, and this carbon then has an amino group up here, and then a double bond to a nitrogen down there. So that's the arginine amino acid. So basically, if you have your polypeptide of phospholamban, you can count the amino acids. You count the first one, the second one, the third one, and this is the amino terminus, by the way, of the polypeptide. And you go all the way up to the 14th one, that will be an arginine. Now if you remove this arginine from the phospholamban, what you've produced is a phospholamban that cannot be phosphorylated by protein kinase A, so it's permanently on. Now basically this mutation is fatal. It causes super inhibition of the SERCA2 pumps, so the SERCA2 function is completely abolished basically, and it leads to hereditary cardiomyopathy. So basically the heart, you're born with a heart which is hypertrophied and you don't live very long. It's very, very lethal. So this seems to be proof of principle that if you have too high phospholamban activity, then it really will stop SERCA2 activity. Okay, and I should also add that you'll always have a bit of constituent protein kinase A activity within the cell, keeping phosphate groups on these phospholamban proteins. Okay, so it's not as though you absolutely need beta-1 adrenergic stimulation to get some protein kinase A activity. You always have a bit. Okay, right. So that overall then is how SERCA2 function is believed to be reduced in concentrically hypertrophied cells. 1, the actual expression is down, and 2, it's over-inhibited by overexpressed phospholamban and also phospholamban which isn't phosphorylated properly.